Italy. So today I'm delighted to be your host in this session. Thank you for joining us. We will get started in a moment. Well, today, we are very pleased to welcome Professor Claudio Avignone Rossa, who will presenting of the topic the electroactivity of the microbial community in the microbial fuel cell for bioremediation. Before we start or we begin, just a quick note, this session will be recorded, so you can find it in YouTube, and we will last around like 45 minutes, followed by question and answers. So feel free to post your questions in the chat or you can just turn on your camera so you can ask directly the professor. So feel free on how you want to post your questions. Now it's my pleasure to hand on over the Professor Claudio Avignone Rossa.
Thank you very much, Hafsa. I will try to share my screen.
Okay.
Can you see that? Oh.
Yes, we can see it. That's right.
Can you see it?
Yes,
okay,
perfect.
thank you.
Okay, perfect. Yeah.
Thank you very much, Hafsa, for the introduction, and thank you to the EBNet for the invitation. Very briefly, I have extended a little bit the idea that I originally proposed for this seminar, because there are many interesting things, or at least things that I consider interesting to discuss. I'm a professor of system microbiology at the University of Surrey in Guildford. I've been there for quite a few years, and I've been working with microbial biotechnology for mainly two branches. One is design of bioprocessing for production of compounds of pharmaceutical industry, biotechnology, etc. And the other part is the use of electroactive microorganisms, your cultures or communities, microbial cells for mostly for bioremediation and depollution, as the title says. And I would like to discuss today how we can understand the composition of microbial community and how this composition is linked, connected to the activity of the community when we are doing some processes such as bioremediation and pollution. And the title, if you want, is not who you are, is what you do, because I would try to show how you can design a microbial community based on the activity that microbial species have. And the of the talk is roughly an introduction, which is very simple, and some of the problems that we need to face. What happens in water, soils, and industrial waste with the complexity of the microbial communities, a little bit of metabolic modeling, and then if we can see, and this is the newer part, let's say, we can see if we can construct metabolic models of microbial communities and how we can apply it, and then some conclusions. Problem of remediation, we all know soils and waters are very dirty. We have been throwing into soil and water many millions of tons of waste. We need processes to clean that water that we have polluted, to clean the soil we have polluted, and there are several different types of approaches for doing that. But some of those approaches are electroactive, and I will try to explain a little bit about that. One of the important things is that we can use single species that are very efficient. They can consume one particular substrate and produce electricity, but these are normally limited to one specific condition. If we have a complex system that is highly polluted with a mixture of many different things, we cannot rely on one single colony, single species. What we need to do is consider a microbial community, because microbial communities present a very flexible collective metabolism that can deal with a very wide range of different type of compounds. Normally, in this polluted water or soil, you find simple molecules, free sugar, lipid, protein, but also very complex mixtures of normally very difficult to degrade compound, recalcitrant compound. So we started very many years ago with this use of microbial fuel cells. A microbial fuel cell, you know, it's a system that utilizes an electrode or an electrode systems onto which a microbial community is attached. That microbial community extract the electrons from substrates, transfer the electrons to these electrodes, and if we connect that electrode into a electric circuit, electron circulates and generates electricity. Therefore, what we have is a system that is using the organic matter present in the water or in the soil to produce electricity. The amount of electricity produced is very limited, it's small, but what you can do is use that production of electricity to clean or to degrade some of the compounds in the medium. So we started using some very naive approach. We said, well, we have compost. You can degrade anything you throw into the composting bucket. We will have an enriched community if we try to figure out what are the preferred substrate for that community. So we enriched the community. We added the complex substrate we wanted to degrade, and we saw that there was some kind of electricity being produced. So a little bit of electricity to light a tiny LED. What happened normally in our system was that we saw that there was an initial period of acclimatization of the microbial community. There was almost no electricity being produced, and after a few weeks, even months, you could see a stable output for the community. So the electricity being produced was like, okay, what we saw is this is acclimatizing, the bacteria in the microbial community getting ready to start consuming. There will be some reaction, metabolic reactions happening that are not really producing any substrate useful for electricity, but in time this will happen. So we tried to study why there were these changes, and we did. And that's to show that we did this 20 years ago or so. The next generation sequencing was not very well accessible at the moment. At that moment, it was very expensive. So we did the good old DGG, the continuous gradient gel electrophoresis, by amplifying the 16S ribosomal RNA genes of the microbial community. And what you see there, and the good thing of this system is that it's very visual. You can see very clearly that some of the species, each band here, which you can see here, each band is a species, right? So this is a gene for one particular species, and you can see that this species that was very abundant at the beginning slowly decreases, reduces the abundance until it almost disappears. Whereas other species that were barely detectable at the beginning, they start increasing and they become more abundant. That means that our system, which was initially not producing electricity, so let's say this part here, the community becomes something that can produce electricity, so those species there. And this is very visual, we can see that clearly, or almost clearly. But the other thing we did is to say, well, what are the different metabolic patterns of those species there? And we saw that we had, at the beginning, we had mostly fermentative bacteria or species. And that means that at the beginning, there was probably only a little bit of electricity being produced because that community was trying to start consuming the complex substrate there. But when they start consuming the substrates, then the electrical or the of species able to transfer electrons to the electrodes became more abundant. Okay, it's only 70%, 60% there are still fermentative, but what you see is that there's an increase in those reducers, nitrate reducers, sulfur reducers, which are normally able to transfer electrons to an electron. So we have a system there that we see is dynamic, it's changing in time. Things are happening. We can measure that. It's very easily measurable. So we can do it. And we found, of course, as I said, it was a very naive approach. We find those species that were most abundant. And we said, if they are most abundant, that's because they are the more relevant. So we found 5 or 6 species that were very active when they were isolated and grown in a microbial fuel cell. We could see that they were producing some power. And we said, well, let's mix them. Let's see what happens if we mix them, if we combine them. So we did combinations of a respiratory species and a fermentative species, or a respiratory and facultative. So we did kind of a combinatorial analysis. What we saw is that none of them, or only a few of them, those combinations produce a much higher electricity or even comparable power to the combination. And even worse, the one that was a combination of all 5 species produced lower electricity than the pure species or combination of 2. So we have a problem because we didn't find what we have. So our hypothesis was not very correct, but we found that we could get some idea from that. One of the ideas is that synergy is lost. So when we separate this species from the whole community, we are losing part of the structure of that community that is promoting a higher activity. What we also see is that there is a competition between different metabolic functions. What happens is that, of course, in a complex community, the flow of substrates and electrons is not linear. So what happens is a very complex network of exchange of substrates, metabolites, electrons, etc., that, of course, some of them are competing. For instance, methanogens are stealing electrons from the electroactive bacteria. So we need to think carefully how we can study this to understand how the community works and make it work for us. So we started manipulating the composition of the community by deleting methanogens, and we saw that under some conditions, the production of electricity improved and the degradation of compounds was much better. So there's a conclusion there, which is very simple. The performance of that community depends on the correct combination of metabolic capabilities, which we didn't need to do the experiment to understand that, but those experiments allow us to create a little more ideas, right? And the idea was how to study the microbial community and how to see if we could extend our understanding of the community by doing a little bit more, let's say, rigorous analysis. So the activity of the different species can be studied by different methods, and what I will try to show you now is how we did that. So very briefly, what happens is we did experiments trying to clean water. This is part of an EU project in which I was involved, the Greener project. And what we have there was a very dirty water coming from an industry producing a dye, a very complex dye. And that water contained also some metals that we wanted to get rid of. And initially, when you do these type of things, the electroactive bacteria can be in the anode, as you can see there, when they are oxidizing organic compounds, but also in the cathode, you can use the electron coming from the oxidation of the substrate to reduce some metals for it. So what we try to do is combine a system with high organic matter content, use it as the source of electricity for the reduction of the metals. In those cases, the amount of electricity you produce is not enough, so you need to apply some voltage. But what we found is that you can reduce the amount of electricity you need to put to convert or to reduce these metals. And those metals can be then separated because they are very nasty pollutants. Right? So the reactions there are a little bit more complex, but the general idea is that we oxidize organic matter in the anode, we reduce metals in the cathode. We use different inocula for this. The inoculars have to be obtained from a system that is working. We know it's working, and we also need to select the right inoculum for both the anode and the cathode. So we found that we could also use an abiotic oxidation of water in the anode. So essentially what we were doing is using an abiotic anode. And we could also see if we could degrade the dye in the cathode. So this DM black here that is indicated here is the dye that was very, very complex, as I said before, with plenty of aromatic rings, etc. That can be reduced in the cathode, and we obtain breakdown products. And we can also generate some hydrogen because this is a reduction reaction. And what you can see, also the metal is reduced here. So we have a cathode that reduces dye, reduces the metal, generates hydrogen, and we obtain cleaner water. And you can see in the table here that the amount of chromium decreased almost completely, and you can see by the color here, this is the water before the treatment, this is water after treatment. You can see there's only a very little amount of the dye here. And the COD, the chemical oxygen demand, was reduced from almost 900 to 200 something. So there's a very interesting decrease in the COD, and there's a very interesting decrease in chromium. So this system can. We also did some studies on the reduction of sulfates in soils, and this is not in water, and this is not electrochemical system. But what we did is we tried to see how the microbial community in the soil was able to reduce sulfate because this is a company working trying to precipitate metals to reduce the amount of these metals in the soil. Those metals, if they are not removed, they will go into the water, and it could be a problem for the pollution. So the company was interested in getting rid of those metals by precipitation in the soil. And we did some studies for the microbial communities in the system for metal and sulfur removal, for metal removal in the biotic microbial electrochemical systems, and sulfate reduction and precipitation of metal. So this graph here shows how the different experiments I briefly showed in the previous slides are analyzed. So what we have there is the study of the composition of the different systems, the different microcommunities in the different experimental setups. And you can see that these are really messy. So there are not many things we can extract from that information, right? The only thing we can say is that there are some genera which are very abundant, some others that are there at very, very low abundance levels. And you can see that there is not much of reproducibility, the variability is very high. So from this information, what we are doing here is a catalog, really. Right? So we have a low taxonomic level catalog. We know classes, we know families, genus, sometimes we know species. It's very useful, we know what's happening, but it's insufficient. What we need to do is try to increase the granularity, the level of resolution of this, the analysis, the type of analysis we do. And those analyses we did using metabolic studies. So we try to study what happened when we apply these microcommunities, electroactive microcommunities in other type of seeds. And in this case, we use glyphosate. Glyphosate is a pesticide, it's a herbicide, which is very controversial. Half of the studies say that it's a very hazardous, dangerous compound. The other half said, no. Many countries have banned the use of glyphosate. Many other countries still use it because it's very, very useful. So in a Newton funded grant, we studied the degradation of glyphosate in microelectrochemical system, and we saw that you can decrease the amount of glyphosate by when you put the water into a microbial fuel cell, and you can see how this microbial community changes. So what we say, glyphosate is degraded naturally in the environment, but what we show here is that you can increase this degradation by using a microelectrochemical system quite dramatically. So what we also found, and that was what we wanted, is to see what happened with the microbial community. The microbial community, they had some, the presence of the species, the dominant species, were all known electroactive genera. Rhodococcus, Pseudomonas, Geobacter are very well known electroactive genera of microbes, and there are other abundant species, Methanobrevibacter, Methanobacterium, that are present in the system that are not electroactive. So the control here was one system degrading glyphosate electrochemically, microbial electrochemically, and the other non-microbial electrochemically. And what we see is that methanogens are displaced by the electroactive species during the acclimatization of the system, meaning that we can improve the degradation of glyphosate by using a microbial electrochemical system, and we are displacing the methanogens, so not we, the community is displacing the methanogens and improving the degradation of glyphosate. So we found that this system is working well, but we also found that the abundant species in the system, there are two different metabolic pathways in these species that can degrade the glyphosate. And it depends if glyphosate is used as a phosphorus source or if it's used as a nitrogen source. As a phosphorus source, glyphosate is converted by a metabolic pathway involving sarcosin, and is subsequently oxidized to carbon dioxide and water. If it's a nitrogen source, it's converted into some intermediate called amino methylphosphonic acid and glyoxylate. So now we have the composition of the microbial community, we know the species, and we have a bit higher resolution because we have a biochemical analysis and a phenotypic analysis. We know also that Pseudomonas are able to catabolize glyphosate via the sarcosin pathway, and this is important for bioaugmentation. So if we want to clean our soils or our water from glyphosate, we can use an inoculum enriched in Pseudomonas. Importantly, the amino methylphosphonic acid and the glyoxylate, that pathway involves some toxic compounds. So some of the intermediates in that pathway, when glyphosate is used as a nitrogen source, are toxic. So if we want to get rid of the glyphosate in the water or in the soil, and we don't want toxicity, we want to use the metabolic pathways that convert the glyphosate to sarcosin and further oxidation. So this is a very interesting result that we have been exploiting a little bit more to try to get rid of other pesticides. The idea here, what I'm trying to transmit to you, is that we can understand what's going on if we increase the level of resolution of our analysis. And that happened when we studied another compound, which is atrazine, and this is in soil microbial cells. So instead of liquid system, in this case, the electrolyte is the soil. So the microbial cell as you can see in the slide, is sorry, I have lost my cursor. Okay, so what you can see in the on the left hand side is a schematic representation of the soil microbial cell, in which the anode is under at the bottom of the system. Then there is soil in between the anode and the cathode, and then the cathode. And you can see in at the right figure a picture. You can see in the soil there are even little plants, little weeds growing there. So this is a real soil microbial cell with using natural soil. This was also part of the Greener project, and that was a collaboration with Mirella Di Lorenzo and her group at the University of Bath. And what we saw is that this atrazine, which is also a nasty pollutant, what we saw is that this is very well degraded by the microbial electrochemical system. But we did some studies, what happened with the microbial communities in time, and in space. So we tried to find out if there were any changes in the composition of the microbial community, depending on where we were in the space and where we were in time. So we started with our initial soil, as you can see there, at time 0. We took the samples for the analysis of the composition of the community, and then our system, we took samples between the electrodes, near the electrodes, on the electrodes, and outside the fuel cell, and we also used a control that was soil without any electrochemical system. And what you see there is that the composition, the microbial composition, the composition of the microbial community changing not only in time, but also in space. And you can say, well, this is little bit messy, but there are clusters, statistically significant differences between the different clusters, meaning that there are different microbial communities. So we can see how the microbial communities change in time, but change in space. And that is important because you want to see how the different microbial communities change and how you can redesign your system to exploit those changes in the composition of the microbial community, because the species will be more or less active for, in this case, degradation of the pollutant. And you can see how here in these plots, what you can see is how the abundance of different species and genera change in time and in the different positions, right? So you can see that between the initial soil and the community in the initial soil compared to the anode changes, the same in the cathode, the same in between the electrodes, etc. So there are changes which are affecting the activity of the system as a whole. And we did a little bit more studies, not only metabolic studies, but also genomic studies, and we detected the presence of very important genes, right? And those genes are genes involved in the degradation of atrazine. And we also found some of the intermediates. So we did a metabolomic study targeting compounds that are known to participate in the degradation of atrazine based on the genes we had. So we were able to reconstruct the metabolic pathways, converting atrazine into the products of degradation until the final production of carbon dioxide. Those metabolites, what you can see with the red dot there, are metabolites that were detected by mass spectrometry. So we did a real map of the distribution of metabolites, distribution of genes, and distribution of species. And that helped us to understand how the degradation of atrazine, or how we can assess the degradation of atrazine by measuring the changes in the metabolite. It was a very interesting exercise, because that now we have an extra level of analysis. We have metataxonomics, metagenomics, metabolomics. We can also extend this to proteomics to understand even more what is going on there. So we can see that there are some species that are potential atrazine degraders. We can see that some species are enriched in the cathode, some species are enriched in the anodes. what's happening there by doing a comprehensive study of the different parts of the microbial community. Now, let me tell you a little bit about metabolic modeling. Metabolic modeling is just constructing a system, a theoretical system, a computational system that allows us to understand the different distribution of metabolic fluxes in a species, or as we want to do in a community. So to construct a metabolic network, a metabolic model, we need to understand the proteomics, metabolomics, transcriptomics, as I said before. With those, we built a map of metabolic reactions helped by literature, or now the last few years, artificial intelligence and machine learning, to understand how these metabolic fluxes are changing in time in a microbial community. So what we do is by doing the metagenomic analysis, we construct maps, networks of the different species in the community. So we need to construct as many maps as models as microbial species we have. Of course, we need to be realistic, and we reduce the size to make it manageable. Otherwise, the computational power we have is insufficient. And one thing we have here is that we use an approach in which the final all the species are consuming a substrate and producing some compounds, and what we consider is that the leftover or the waste of one of the species is used by another species as a carbon source, etc., etc. But the final electron acceptor will be an electron. So what we're trying to do to construct is a metabolic network of a microbial community that eventually will yield the electrons to an electron. In reality, what we have is that not linear, as I said before. What happens is that there is an intercommunication between the different species. I think that map has been spoiled a little bit, but anyway, what I'm trying to say here is it's not linear. It's a network of reactions, and there are too many interactions between the different species. So what can we do with this metabolic model? Well, we can identify which are the species with higher activity based on the computational analysis and the simulation. We can identify which are the essential metabolic activities, how the different species act, so what's the division of labor between the different species, how this system, the complex ecological system interact, and how we can determine the different relation between the different species, if they are commensalists or mutualistic or competition, etc., etc. Well, what we can do, as I said before, is try to find a reaction, a series of reactions that ultimately transfer electrons to an electron. This is the most difficult part because that reaction is not a biochemical reaction, not a metabolic reaction. So it's very difficult to put it in a metabolic map. However, we try to find out which are the metabolic pathways that present higher activities and how some of these interactions can maximize the activity. So if we have 4 species, for instance, this is purely theoretical, we see that this is coming from the original work with the DGG analysis. And what we see is that some of the species will be growing and will be producing electrons. And we need to create a map in which we can see that those interactions and see if we can reproduce the production of electricity in the model. Once we have that, we would be able to create a synthetic artificial microbial community that is able to produce electricity by inoculating a microbial fuel cell. So we can see the potential interaction between the species, but this is not giving any details because this is a qualitative model. What we want to do is create a quantitative model, but this is something we are working on. So there are many different problems to do this and difficulties that we are trying to solve. This is something that is a metabolic model is never final. It's always evolving, and so it's a continuing process. You will never find one metabolic model that said that's it. This is the one, right? So there are several problems that we are facing, but we believe that we're going to be able to construct a good microbial electrochemical model for this type of system. So in the last 4 minutes that I have, I will tell you something about what we do. So what we do is we study metagenomics, metataxonomics, transcriptomics, all the omics you want. We construct a metabolic model. That metabolic model we can analyze and see what's happening inside it by doing this flux balance analysis. We understand the composition, we understand the metabolic capacity of the system, what are the functions, what are the activities, and that will allow us to understand how this microcommunity work, how can be optimized, and how we can create an artificial community. Now, I'm not a microbial ecologist, not even an ecologist. So what I'm going to say now in the next 2 minutes, probably if there's an ecologist in the audience, they will complain. But this is my view. So, and a very simplistic analogy of a micro community is an orchestra, right? So what we have there is we can study an orchestra. We have the different type of instrument there, the family of the woods and the brass and string family, right? So we go there, we make a catalog, and we know what type of instruments we can see in an orchestra, right? That will be when we do metataxonomics. We see is there are 3 violins and 10 horns and blah blah, right? So that is only telling us which are the species present in the community and what are the different compositions for different conditions. If you want I don't know, a chamber orchestra, you will have only 9 or 10 instruments. A string quartet will have 4 instruments. A symphonic orchestra will have 120 instruments, depending also in the piece you're playing, the composition you're playing. So it depends. Even depends on the conductor. If the conductor has some ideas about how many brass should be there, well, he or she will apply. But we don't know anything. So we see the catalog of instruments, but we don't know how the orchestra is going to sound. Right. So what we need to do is understand what's happening with each instrument. So we need to understand the capabilities of each of the instruments. We need to understand how they interact between them, the first violin and the violas and the cellos, you know. So we need to understand how they link together. So they will be a little bit like studying metatranscriptomics, metabolomics, biochemistry, metabolism. So we know roughly how these instruments interact between them, and we can imagine how they're going to sound. This is the function of the composer, right? So it is also known that Beethoven, being deaf, he could also in his brain he could understand how this orchestra was going to sound, right? So this is the function of the composer. So the objective we have here is try to make a model that will tell us if I put these instruments together, how is this orchestra going to sound? So if I put these microbial species together, how is this community going to work? Right. So if we continue this analogy, metabolic modeling would be try to build a theoretical community to simulate the performance of the real community. Right? And we can also try to predict the performance under different conditions. Now, so I think it follows from what I think that, well, it's an analogy, very simplistic, and as I said, not being an ecologist or a musician, well, maybe those people will complain about that. Anyway, the other problem we have is sometimes some of the instruments or the species are not available. So what happens if we don't have a species in our native microbial community? Can we replace those activities with species that have the same capabilities? Right. So if I don't have a Pseudomonas that can degrade toluene, can I use other Pseudomonas-like species that can do that? Okay, so that is like, if we don't have our instruments, we can try to find out how to replace that. And that has been done for many, many years. People trying to reproduce sound of different type of real instruments by using something that are not real. And this is what I call a synthetic community. Synthetic community is a community that will have similar activity to the natural native microbial community, using species that are not the same. Right? So, as a conclusion, is that, well, if we need to understand and we need to know what the metabolic phenomena present there are in the community, what are the mechanisms. This is the only way of solving and understanding how complex these bioelectrochemical systems are, and then try to make them work for us. We need to understand, I'm sure we all are clear about that, multi-omic analysis is very, very important. We need to understand at the highest level of resolution possible the different tools we can use, because otherwise we cannot elucidate what the community can do. We are doing metabolic models of microbial electrochemical systems, but it's still early. I think there is only one or two metabolic models of single species that can be used, but still they are not very efficient. And we can try to invent or create synthetic microcommunities, because this is a very powerful tool for our design of our bioprocess, be it degradation of pollutants, or even production of some things, or even food production. And we are still working. So I cannot read the list of all the collaborators. This is just a summary of all the collaborators. And this is a list of the research projects that have been supporting our research in Surrey and other places. But we have been working, as you can see, almost 20 years now. And I have been funded by all these different research funding entities, including BES and the predecessor of EBNet, I believe, is AD Network. So the networks in industrial biotechnology from BES have been very, very helpful in our research. And this is all I wanted to tell you today, with only 4 minutes late. Thank you very much.
Okay, thank you so much, Professor Claudio. Such wonderful information and insights about the microbial fuel cells. If you have any questions, type.
And the general idea is to design a community, a synthetic community, with functions, with targeted functions, with the functions we want them to do.